One of the best ways to diagnose breast cancer early, aid in treatment decisions or predict treatment response is to use cancer biomarkers. A biomarker is a substance in a person's blood, urine or other body fluids. It can also be found in or on the tumour that is produced by the cancer tissue itself or by other cells in the body in response to cancer. Researchers use biomarkers to provide insights into a patient's individual cancer. HER2-positive early breast cancer molecular biomarkers are a research area for Associate Professor Alice Prat. Associate Professor Prat is the head of medical oncology at the Hospital Clinic of Barcelona in Spain and was an international guest speaker at the latest Breast Cancer Trials annual scientific meeting. We sat down with Associate Professor Prat to ask what the biomarkers being researched in HER2-positive early breast cancer are and why are they important.
So HER2-positive disease represents around 20% of all breast cancers, so 1 out of 5 women develop this type of disease. On one hand, we have good news, that is that we have very good effective treatments, chemotherapy and drugs that attack the HER2, and this is great because this is a tumor type that if you don't treat it is quite aggressive. But at the same time we know that this is not enough. We know that we are on one hand over-treating some patients and on the other hand we are under-treating some patients. So we need biomarkers, we need some sort of determination or assay or test that allows us to identify the different groups of patients within HER2-positive disease. And this is what we and others are working continuously. And today I think we have several tests, several biomarkers that are starting to subdivide this group of patients into different. On one hand, one is what we call the molecular subtypes. This is a biomarker of 50 genes that helps us divide HER2-positive disease into 4 different groups. And today we have substantial evidence that these different groups behave differently and respond differently to the standard of care, to the treatments we're using. In particular, there is one group of patients with this biomarker that seem to benefit a lot from drugs that target the HER2. So this is the group of patients that potentially in the future, if we do the right clinical trials, we might be able to get rid of chemotherapy. So this is good news, but still we need to run the studies in order to test that, because this is just at this point under clinical research. This is not standard practice. The other biomarker is the immune system. We know that there is a substantial proportion of patients that they have HER2-positive tumors, and these tumors are what we call inflamed. They have a lot of inflammation, they have a lot of cells that come from our own immune system that are detecting the cancer and are trying to fight. They are not winning, because if they win, there's no cancer. So that population of patients, we know that on one hand they have a better survival, and this has already been shown. So potentially this biomarker could help us also to de-escalate therapy, because they have a better outcome. We could de-escalate chemotherapy. And at the same time, this is a group of patients that if they don't respond as we want, potentially immunotherapy in the future could be a strategy to pursue. So overall, as you can see, we are starting to subdivide HER2-positive disease. This is good news. We need to run the trials to really have enough evidence to say now it's the standard of care, but it's looking very promising.
So Breast Cancer Trials currently has 2 immunotherapy clinical trials open, Chariot and Diamond. Do you see this being a big part of the future of specifically HER2 breast cancer?
Well, I think in general, immunotherapy in breast cancer will come. It's slowly, but it's coming in all subtypes, not only HER2-positive disease. The good thing about HER2-positive disease is that we have many drugs that are active and they are not immunotherapy per se. And therefore the immunotherapy that is coming, we'll see where it falls. But I do believe that HER2-positive disease will benefit from immunotherapy in the future.
So these molecular biomarkers, how can they help to prove a de-escalation strategy is safe and beneficial for patients?
So we have accumulated a lot of evidence in the last years, and this has been thanks to tumor samples that we have analyzed from trials in HER2-positive disease. And I think the biomarker, the molecular subtypes, I think now we have enough evidence to say that we feel reassured and we feel certain that we can select these patients with this biomarker and run a trial where we can de-escalate chemotherapy. And instead of treating these patients with 6 months of different chemos, potentially we can treat these patients with 3 months of one chemo only, together with the antibodies against HER2. The trials are designed, it's just we need to run it. We need to run the study, show that by selecting with this biomarker that we are assured, and show that they have a great outcome. In a few years, I think we will be able to conclude that now is the standard of care. We can identify that population, and if they have this biomarker, treat them with less chemo. And that, from a patient perspective, that would be great news, because we need to de-escalate systemic therapy in this group. Another area of de-escalation is also surgery, and this is something that these tumors are responding so well to standard therapies that when we give these drugs before surgery, in a high proportion of patients, there's no tumor left, which is great news. But at the same time, many patients, after 2 weeks of surgery, they come to my visit and I tell them, "Oh, it's great news. You didn't have the tumor." And then the second question that they ask is, the first is, why did we do surgery if there was no tumor left? And of course, today we need to do surgery, no doubt, but potentially there is a subgroup of patients in the future that maybe we don't do surgery. And that would be also amazing news, I think, for patients. And in HER2-positive disease, we have an opportunity to prove that.
Is there any clinical trials in this area that you're working on that you're quite excited about?
So we are about to open a study in Barcelona at my center. It's a pilot study because we want to go slowly into that direction, where we will select patients with HER2 positive and with a particular biomarker, the molecular subtype, and show that if we treat them with standard therapy and they have an excellent response by MRI and a biopsy, we are not going to perform surgery. This is going to be a study of only 18 patients, but it's just a way to start opening that door, which is to de-escalate surgery. There's another trial in the US currently in HER2 positive and triple negatives with a similar strategy. They don't use biomarkers, but this is a multi-center trial that also, if they achieve a response with MRI and a biopsy, they don't undergo surgery. So there's already some trials going that direction. So I think it's worth keeping an eye on those results.
That was Associate Professor Aleix Prat, Head of Medical Oncology at the Hospital Clinic of Barcelona. If you'd like to learn more about breast cancer trials or you'd like to support our life-saving research, follow us on social media or visit our website at breastcancertrials.org.au.